The phototransduction in the rod cells of a retina. This process begins when a photon enters the eye and hits a thin layer in the eye called the retina, represented by the orange. On the retina, the photon hits the rod cell and is absorbed by the disc of the rod cell. Inside the disc on the rod cell is what actually absorbs the photon. It's a photopigment known as rhodopsin. Rhodopsin is made out of two separate parts, black representing the opsin and orange representing the retinal. But when the retinal absorbs a photon, it rotates from its 11-cis retinal into 11 transretinal. This causes the retinal to rotate and straighten out. When it straightens out, it then creates a change within the whole of the rhodopsin. This causes the rhodopsin to become active. The next component of phototransduction is the transducin, also known as the G protein. The G protein is a trimeric protein made up of 3 units, the alpha, beta, and gamma. When inactive, the G protein is bound to a GDP, also known as guanosine diphosphate. When the rhodopsin has changed due to it absorbing a photon, it can now activate the G protein. It also switches out the GDP with GTP, guanosine triphosphate. The GDP then takes the alpha subunit of the G protein and goes and activates a photodiesterase, also known as PDE. Once PDE has been active, it starts making cGMP into GMPs. To understand why this is important, we must take a step back and look at the charge of the cell. The charge of the rod cell is different from most cells in the human body because in a state of rest, the rod cell is depolarized. It is depolarized because the cGMP keeps open a cGMP channel, allowing sodium and calcium ions to flow through. When the cell receives a stimulus and the PDEs start changing cGMPs into regular GMPs, then there is no longer any cGMPs to hold open the cGMP channels, causing sodium and calcium to no longer be able to flow freely through. Because sodium and calcium ions can no longer freely flow into the cell, this changes the charge of the cell. Because the charge of the cell starts changing, the potassium voltage-gated ion channels then open up, allowing potassium to freely flow through, and the cell starts to hyperpolarize. When the cell hyperpolarizes, that signals to the cell to start sending less transmitters. The less transmitters from the rod cell to the brain signals the brain to help us see. This process can only happen for so long until the rod stops receiving a stimulus. When it stops receiving a stimulus, then the protein kinase G comes in and inhibits the rhodopsin from activating any more G protein. The next process is when the enzyme guanylyl cyclase starts converting GMPs back into cGMPs. The last step in the process is the activation of the sodium-calcium-potassium exchange pump. It exchanges the sodium, potassium, and calcium until the cell is back in its state of depolarization. And that is the phototransduction in the rod cells of the retina, which allows us to see and interpret light.